She is the speaker today.
Screen share karo.
Okay. Share screen. I do desktop, right?
It should be. Oh yeah, it's coming. Oh, desktop, but yeah, good.
Is this good?
Yeah, full screen and presentation mode. You see presentation mode? No, yeah, also on the right up. Okay, good.
And now. Is this good?
Okay, 1 minute. Najifah is a brilliant student, as you see, and she is focusing for medicine. I am hoping that she will be very, very successful doctor, and that's why I asked her to present this thing. Okay, the students, everyone concentrate. Okay, start.
Okay. Hi everyone. My name is Najifah, and today I'll be doing a seminar on cell, the unit of life, coronavirus epidemiology, and prediction and control from the research point of view. Okay, 1 second. So before we begin, here's an agenda of what we will be doing today. So I split this seminar into 2 sections. The first is cell biology. So we'll be just reviewing some basic information about what is a cell, cell theory, variations, components, DNA, genes, cancer, and viruses. And then we'll be going on to an application, which is the epidemiology of our current virus crisis of COVID-19, looking a little bit to what the field of epidemiology says about this virus, some background, the current situation, facts and figures, some patterns, and obviously some symptoms, transmission, and risks factors. So without further ado, let's begin. So what is a cell? The website definition or any textbook definition would say that the cell is the basic structural, functional, and biological unit of all known organisms. So the cell theory basically states that all living things are made up of cells. Cells are the smallest unit of life, and cells are made from pre-existing cells. What this means is that when a cell, like the cells we have in our bodies, they're all from old cells which have divided and created them. And also what this means is that the cells is the smallest thing that can carry out all of life's basic processes. So I have a question for you guys. How many cells do you think are in the average human body?
40 trillion cells.
Yeah, trillions of cells.
Okay. That's actually really close. There's 37.2 trillion cells in the average human body. Obviously, this changes according to your age, your body size, and your gender, but on average, it is 37.2 trillion, so good job. So there are multiple different types of cell in the body. These are just some examples. So we have the red blood cells, which, so in terms of blood cells, there's 3 different types. We have red blood cells, which transport oxygen, white blood cells, which fight infection, which we really need right now, and platelets, which are used for blood clotting. So whenever we have like a wound and we start bleeding, the platelets help clot up that blood so we don't lose too much blood. And then we have fat cells, which help store fat that can be used for energy and form adipose tissue, which our body uses to produce hormone, blood pressure, and do cell signaling. And then obviously there are some bad cells which can form in our bodies, such as cancer cells, which is an abnormality yet again, which we will I will continue to discuss that as we continue. So when it comes to organization within our body, it first starts off with a cell, which will then form a tissue, which then forms organs, which forms an organ system, and then eventually an organism. So this is a diagram of a cell, and now we're just going to be going into what all of these little colorful things are. So these colorful structures are called organelles, and they're structures within the cell that perform specific functions. So it's like a really big machine within our bodies that have tiny, small machines within them. And basically, if you can see here, it doesn't show that well, but there should be a membrane here. And basically, that's the cell membrane, which is the outer boundary that controls the passage of materials. And here, all the white space here, that's not just empty space. It's actually a jelly-like fluid, which the organelles can float on. And it will contain these structures called ribosomes that will help make proteins. And in this fluid, a lot of our body's chemical processes will take place. So these are some key organelles within our cells. So I'm just going to explain some of them. Well, all of them here. So this is the nucleus. The nucleus stores DNA, which contains genetic information and controls our cellular activities. So inside this orange nucleus is this purple part, which is your nucleolus, and this will help your body or the cells make proteins. And then this is the mitochondria. It is what is known as the powerhouse of the cell. It will help the cell create energy. This is the endoplasmic reticulum, which will manufacture and ship cellular products, and this will help protein synthesis. This is the Golgi apparatus, which organizes and ships cellular products, so these two will work hand in hand. And this is a lysosome, which contains digestive enzymes, which will help break down waste materials. And this is a vacuole, which is used for storage of water and etc. So the most common thing in biology, which we begin to look at, in the beginning, we tend to focus a lot on this. It's the difference between an animal cell and a plant cell. So as you can see, they look really different. They are, but they're not as different as the pictures make them seem. The only thing that's really different is that the animal cell have these structures called centrioles, which they use to basically help cell division and organize the genetic information. And a plant cell will have primarily, it will have a larger vacuole for storing water. It'll have a cell wall to maintain its rigidity, and it'll have chloroplasts, which are these green thingies over here, which will basically help it convert the sun's light energy into sugar so it can eat. So within the nucleus, you have your genetic information. So it's where DNA, which is our genetic information, is stored. And DNA is not only our genetic information, but it also provides sort of like a blueprint for our cells to perform their functions. So if you can see from this diagram, it's a very good diagram. It shows that this is a chromosome, which is a tightly wound DNA. And basically, this part is a gene, which is a small section of DNA that will code for a particular trait in a person, like freckles. And again, DNA is the material that makes up genes and chromosomes. Stretched and unwound, it looks like a double helix. So if you could see, it looks like a ladder. So obviously, there are abnormalities that can happen within our cells. So this is called cancer. So cancer is the uncontrolled growth of abnormal cells. Oops. It will form lumps called tumors. And what's interesting about cancer cells is that they don't divide properly. So what this means is that the daughter cells that do form, they contain abnormal DNA. And basically, normal cells, they stop when there is abnormal DNA in order for the cell to either completely destroy itself or try to fix its abnormalities. But since cancer cells do not divide properly, they don't wait and meet the checkpoints, which would ensure that they have the proper DNA. So they just keep going with this abnormal DNA, and that's what causes malfunctions. And cancer can be caused due to random mutations that can happen, but these can also be as a result of carcinogens, which are cancer-causing agents. So like too much exposure to UV light or radiation or even tobacco smoke. So in the lab I work at, we focus on a special type of cancer called sarcoma. And sarcoma is a really rare type of cancer that you don't really find. But when you do, the cases are pretty severe. And um, basically, it's a cancer of the connective tissue. And what we do at this lab is that we extract the cells from patient tumors and then grow them ourselves in our labs to test out different drugs. And so recently our lab found something interesting on a biochemical pathway that's basically a series of reactions that happens within our body. So this pathway is called the P13K biochemical pathway, and it drives glucose uptake and is frequently a mutated pathway in cancer. So what they found is that it showed through different cases that two common, really like abrasive sarcomas, they actually rely on metabolic reprogramming, which is the ability of our cancer cells to alter their metabolism in order to support the increased energy requests due to continuous growth and growth through this pathway. So they also discovered that basically a drug called simvastatin can actually inhibit this pathway to make the growth like almost stop, like decrease to the point where it's really close to stopping, but we're not quite there yet. So now moving on to viruses. Viruses are non-living, obviously. They're an infective agent, meaning that they can't really, meaning that they're pretty much parasitic. They can only multiply within the living cells of a host. So basically what this means is that viruses require a human or an animal in order for them to be able to colonize the body and extend. So what viruses generally are is that they're genetic material, whether it be the DNA that we talked about earlier or transcribed into RNA, wrapped in a protein coat. So there are multiple different types of viruses. This is just one of them, and this image shows it injecting its genetic material into a cell, while this is just another one here. And coronavirus actually looks more like this. So types of viruses that we found. So this is the helical shape. So as you can see, this is made out of RNA. It'll have this type of capsid, which is basically a protein coat that surrounds the genetic material. This is polyhedral, which was found in the adenovirus. So this is actually a DNA virus with a glycoprotein type of protein wrapped around it. And then this is spherical. So this is the influenza viruses that we usually find in common colds. And also this is the shape that COVID-19 actually takes. So there's the protein coat and there are glycoproteins attached, and there's RNA in this one. And then this is the complex bacteriophage, which has a head, tail fiber, tail sheath, and it's a DNA virus. So moving on, viruses tend to have different life cycles. So there's two different things they can, two different cycles they can go through. So the first cycle would be the lysogenic cycle. So if this is the cell, this is the original cell. If the bacteriophage comes and injects its DNA in, the phage DNA will circulate, and then basically what happens is that it can either go to the lysogenic cycle, in which nothing really, like it stays, the virus DNA stays dormant even though it replicates, or it could go into the lytic cycle where it can lyse. So if it goes into the lysogenic cycle, then from this diagram, you could see that the phage DNA integrates into the bacterial chromosome, as you can see here, and then the bacterium will start to reproduce. And what will happen is that you'll have multiple copies of this type of genetic code with the bacteriophage's viral DNA in it in multiple different copies so you would have it in multiple different cells. And then cell division produced population of bacteria infected. And then basically, occasionally a prophage exits the bacterial chromosome, initiating a lytic cycle. So it would go into this cycle, which is the harmful cycle. So if it goes into this cycle, then what happens is that new bacteriophage created on proteins within the cell, because the viral DNA will now inhibit and tell the cell to create more of these viruses. And when they do, the cell may lyse, which means it may burst, which will release more of this virus into the body. Okay, so now we're going to be talking more about the epidemiology of COVID-19. So before we begin that, we have to go through what is epidemiology. So generally speaking, epidemiology is the study of the distribution and determinants of health-related states or events in specified population, and the application of the study to control health problems. So this is kind of a long definition, but if you look at the word and the Latin roots, you can really tell what this means. So epi means on, like on something, demos means humans, and ology is the study of. So it is literally the study of what befalls upon humans, which is obviously disease. So usually epidemiologists use something called the epidemiological approach, which is showed by this image. So in the beginning, a health problem or burden is identified, and then they determine influencing factors or causes that cause this problem. And then there's a selection or development of effective intervention or measures. And then we do an implementation of the selected intervention or measure. And then we evaluate the operation and the results. So like we evaluate the effects on the community. And if the problem is still not solved, we just continue this cycle over and over again. Epidemiology, a lot of people do not really know about this field, but it's a very important field because it not only stopped, like the most notable thing it stopped is that when smallpox was very common amongst individuals, they used epidemiology to track down cases, track down what started it, and put an end to it. And also they used epidemiology to stop cholera outbreaks too. So now moving on to the spectrum of disease. Most diseases, almost all diseases follow this spectrum. So in the beginning, you have a stage of susceptibility. So this is when you have appropriate exposure to factors or agents that cause disease. Actually, before I start explaining this, I should explain that in order for you to have disease, there's three factors involved. There's an agent, there's a host, and there's the environment. The environment will bring the host and the agent thing together. And if there is a susceptible amount of agents, they will penetrate the portal of entry of the host and thus become viral infections. And so basically during the stage of susceptibility, again, there's an appropriate exposure to factors or agents that will cause the disease, so exposure. And then you move on to the stage of subclinical disease. And this is basically when you have pathological or chemical changes within your body. And this can be asymptomatic, in which you will have or show no symptoms, or you could start to have symptoms, but it's usually at the end we detect any symptoms, and that's when we have our diagnosis. And then it's the stage of clinical disease, which is again marked by the symptoms. And then finally, it's the stage of recovery, disability, or death, depending on multiple different factors. So now moving on to COVID-19. So COVID-19 was caused by a new type of coronavirus. So in the media, they talk a lot about the fact that we call this the coronavirus, but we shouldn't really call it that because coronaviruses are actually a large family of viruses. So it's more of a general term. and coronaviruses can transmit from animals to humans. And basically, this is actually named the SARS, so severe acute respiratory syndrome coronavirus 2. And this is a type of beta coronavirus, such as MERS and SARS. So SARS is, again, the severe acute respiratory syndrome, and MERS is, I believe, the Middle Eastern respiratory syndrome. And basically, all of these had origins in bats. So when they found what started coronavirus in China, they did find that it did originate in bats, just like MERS. And as you can see, diagram of what the coronavirus looks like. So again, it originated in Wuhan, China, and at first they thought it was a different type of transmission, but then when they started looking at different cases and how people were getting the virus, they found that it was actually more likely to be person-to-person spread. So now in global terms, it's actually community spread. So community spread means that some people have been infected, but the exposure time and place is still unknown. So it generally takes 5 to 6 days to show symptoms, but usually they also say, just in case to wait 14 days. So that's why I wrote 5 to 14. So the thing that's different about COVID-19 is that it's actually the first pandemic, which is the disease that has nationally or globally spread, known to be a coronavirus. So there were 4 past pandemics that happened, and they were all influenza viruses. So most of the research and guidance that we actually have today, it's based on these influenza viruses. So we don't really have anything specific to test. So this is just a graph, not a graph, sorry, an infographic about like the number, like the cases in Canada. So this, I actually got this today, so April 18th. So as you can see, there's 32,412 cases, and there's more, like the darker the color, the more severe the situation is. So the more number of cases there is. So as you can see, Ontario, Quebec, Alberta have very high amounts. And if you look here, this is a graph. It's called an epigraph or an epidemic graph. So usually there are bars here, but for this, it's just a curve. So as you can see, it's a relationship between the number of total cases and the reporting date, where this is the first case right here. And basically, as you can see, the has yet reached its peak, so it's still increasing. So again, some more facts. Number of people tested in Canada as of this morning, so 11 o'clock, 516,216. Confirmed cases, 32,400. Probable cases, we're looking at 12, and deaths, 1,346. And then this is more statistics worldwide. So number of COVID-19 cases worldwide as of today is 2,254,388. Estimated mean infection rate is 2.5. But a lot of the times these numbers are not completely representative, because there are some countries which, because of political reasons, have decided to skew the numbers. So this is just a further analysis of the curves for Canada, different provinces of Canada. So as you can see, Quebec has the most, and flatter lines equals to slower growth equals to flatter curves. So what we want is we want the curves to start to become flat or plateau. So that would show that no new cases are like occurring, no new cases are discovered. But even though this graph looks like it's going to plateau soon. According to the Canadian data websites, the cases continue to increase every day, and it's not going to flatline because it hasn't reached its peak yet. So again, this is just another thing about flattening the curve. So a lot of the times, this type of, when it comes to disease and pandemics, our attitude plays a big role in it. So as you can see with person, he is very ignorant and he just states that, oh, it's like a cold or flu. And obviously the peak would be much higher and there would be a higher number of cases, while if you don't panic, but you do take cautionary measures such as washing your hands and not making direct contact, the peak is much lower. So generally speaking, pandemics of respiratory diseases have found to show a similar pattern. So the first phase would be the investigation phase, where cases begin to show. The second would be the recognition phase, where there is a significant amount of cases. And then is the initiation phase, where person-to-person spread is becoming more and more common. And then it's the acceleration phase, which is basically when the rate of cases start to become exponential, and the illness is at the end, the acceleration phase. And at the acceleration phase, so if you look at the curve, the acceleration phase would be when it's going upwards. So then when it reaches the point, it'll start decelerating again, and that's when you'll see a decrease in illness. So symptoms of COVID-19, as like, it's more like a common cold, the symptoms for mild cases. So it causes infections of nose, throat, and lungs, common cold symptoms, fever, tiredness, dry cough. In severe cases, obviously, the symptoms are much worse. So in terms of transmission, respiratory droplets from cough or sneeze of someone who is infected, close or prolonged contact, touching something with virus, then making contact with any type of portal of entry, and person-to-person spread, which is the most common way of this being spread. So in terms of risk, people 65 plus are shown to have the highest amount of risk, and people with underlying medical conditions such as diabetes, heart conditions, and also any type of lung disorder. Also, people with compromised immune system tend to be more vulnerable. So moving on, in media, they tend to use the terms quarantine and self-isolation interchangeably, but there's actually a big difference in them. So quarantining is for 14 days exactly, which would be how long it takes to see if there's symptoms of the virus or not. So this is for people who initially show no symptoms, or if they're just returning from travel, or they went in close contact with someone with a suspected case, versus some, which is basically a prolonged period of staying at home. And this is when you're diagnosed with COVID-19, you're waiting for results, your symptoms are mild to severe, or you got contact with a confirmed case, or you're returning from travel with confirmed symptoms. So in terms of what you can do, you can stay home when you're sick. You can wash